Bonjour. Hello. Good morning. Je m'appelle Paul Kennedy. My name is Paul Kennedy. Je suis l'animateur d'une émission radiophonique qui s'appelle Ideas. My name is Paul Kennedy. I'm the host of a radio program called Ideas on CBC Radio 1. Thank you. It has been my pleasure to be the host as well of the Friesen lectures for now, I cannot believe it, more than one decade, which is amazing. When one first accepts a job like this, it's always difficult to say, welcome to the first annual lecture, because you never are quite sure there will be a second. Well, this is the 11th, I believe, and it is a great privilege to have been at all of them. A privilege because in my job, it's good to keep on top of developments, not just in science and the sciences and medicine, but in basically all fields of intellectual endeavor. And by keeping on top of what's happening with people who win this Friesen Prize, I'm certainly on top, or at least I can give myself the delusion of understanding that maybe I know what's going on in the field of medical research, because the laureates in this prize have been some of the top people in medicine, not just in Canada, but in the world, and that tradition continues very much today, as you will soon hear. My job today is basically to be MC, to keep the traffic rolling, and the first thing I will do in that context will be to introduce the person, the angel, I would say, who is in some sense responsible for the award from the very beginning, Dr. Abby Angel.
Well, thank you very much, Paul, for that 6-second introduction. Very generous indeed. I enjoyed reading and reviewing the registration list and discovering that about 70% of the people here are coming to this for the very first time. So a lot of people in attendance really don't know about Friends of CIHR, the Friesen Prize laureates, so it's worthwhile just spending a millisecond or more just to acquaint people with a bit of background. The history goes back quite a long way, actually, and I'll begin, of course, by thanking the University of Ottawa, Mona Niemer specifically, for being such a fine host over the years, collecting the intellectual strength of the university to come listen to supreme scientists and leaders, and of course, today is no exception with Dr. Janet Rossant, who will be introduced more formally. So. In just a second by Alex McKenzie. I guess this goes back quite a few years. I guess 16 years ago when Friends with CIHR was established, the precursor institution was Friends and Alumni of MRC. It was Dr. Henry Friesen's prescience and vision to realize that it was important to reach out to his communities, both in the free living community and society and the community of scholars in the country, to invite them, have them participate in an annual basis to understand what was going on in science and to understand science as it is being practiced, because there's a big distance between the frontier of science and the knowledge base in the community. I had the great privilege of being part of the transition from MRC to CIHR. Alan Rock, as president of the University of Ottawa, was there as well, promised us $1 billion at the time, year 2000, and I think we've reached it now and signed, which is wonderful. So I want to pay tribute to Professor Rock. And so the program continues. We're in our 11th year. Last year we had Sir Paul Nurse and very distinguished people. This year we have our own Canadian measure of excellence, vision, leadership, and science, Janet Rossant, and we'll hear from her directly in her work in stem cell science and developmental biology. I'd like to point out that Friends of CIHR over the years has evolved to a significant extent, apart from outreach to society, support of young people in their careers, communications on behalf of all, and support of CIHR in its vision and principles. We've also espoused and developed links with other venerable institutions, explicitly the Banting Research Foundation, and Kathy Whiteside is here, president of that, and she has a fine banner showing, and also RCIS, Royal Canadian Institute for Sciences, and we have the president here, Peter Love. Just wave at the audience. You see the back of his head, it's gray, front is prettier. And the past president, Hella Tosin. Thank you very much for coming, participating. And these institutions, 100 years old plus, have joined us in mounting roundtables and symposia, which go on in tomorrow's program. Now, I think I'd like to just continue on with a brief slide. You've seen this, of course. It prompts me to mention the fact that we can't do very much in the absence of sponsors and supporters, and we have distinguished representation here. Rosie Goldstein from McGill University. Rosie, just say hello, is with us and has been a strong supporter. University of Manitoba, another, the home of Dr. Henry Friesen. And Grant Pierce is here, a member of our board. Grant, thank you very much for coming. It's interesting to know why Dr. Rossant was given the award. And I thought I'd share the citation. She's seeing this for the very first time, but this will be presented at an award dinner tonight, and it tells you about what she's done in a sense and why she's being recognized for important discoveries in developmental and stem cell sciences and visionary leadership in health research. You know her from her publications, but now you'll learn from her in person, and she'll give the talk that's mentioned in your programs. Now, just the principal activities of Friends of CIHR, for those of you who haven't heard of us before, one of the signature activities of Friends is the Friesen Lectures. Now it's Janice Rossant. Here she'll speak at the University of Ottawa. Wednesday at McGill University, another host institution for an institutional visit. And then following that, University of Manitoba in 2017, UBC, U Calgary, and U Alberta, Edmonton. These are sponsoring institutions, and we thank them publicly for their support, but also for hosting science and the principles of supporting science for society, which all you participate in any event. Then our institutional partners, as mentioned, the Banding Research Foundation, the Royal Canadian Institute, and the Canadian Society for Clinical Investigation, which we have been affiliated with for a long time, right from the beginning. And just to point out that as the annual FCIHR lectureship in collaboration with CSCI in Toronto, Dr. Linda Rabinick, the Canadian Academy of Health Sciences new president, and we appreciate that link as well. Roundtables, as mentioned, the role of discovery research in health Canadians. Does Canada have too many PhDs in trust and science is what we did last year. What we do is immortalize our presentations by publishing books from manuscripts provided by people like Janet Rossant. This is book 4. She will be contributing along with Sir Paul to book 5, a real keepsake, and I hope some have been distributed at the table for those who were here early enough to get the prize. We publish our roundtables. We want to promote the fact that policy is important in health and health research. This shows us the publications from last December's roundtables here, and then the latest one in Alberta, Trust in Science. Now, this organization is thin, so to speak. And here I'm featuring the FCIHR administration. Like all of you who present talks, you always at the end show a picture of the lab. This is the lab. On your right is me and Christina Castelvi on the left, and I want to pay tribute to all the work that she does to make this a success. Now we need to move on in the program, and I'd like to ask my colleague, member of our executive, Alex McKenzie, who is here representing the university, but also has the honor of introducing the guest of honor.
Thanks so much, Albie. Do-si-do here. That's great. Thanks. That's great, Albie. I just have to say there's chairs down at the front for those that want to sort of make their way down in this interregnum. I have 40 slides I'm going to be going through, so you have no slides. Yeah, I'm here as University of Ottawa representative, which would be a surprise to University of Ottawa, perhaps, but also as a secretary to FCIHR. And having me as a secretary to anything, I perhaps you need a bit of a rejig, but it's an honor to be on the executive for sure. So Janet, and I'll keep this brief because I know you'd like to keep it brief. I first met you, and you don't remember this because we've had this discussion, when you were still at Brock. We were doing an external review at the University of Guelph for some of the porcine program, and you were clearly the brightest, most au point member of the committee. I found I was a young, sort of ambitious professor at the time. I was a little taken aback by the clarity of your thought, etc. And but just struck me as first impression. In case you were worried about with me, made a very good first impression. And then on to actually sign it with, I guess Alan was the one that sort of brought you in and did phenomenal stuff with mouse development. For my mind, all the premier journals are great, but if you're publishing in development, I love that journal and the beauty and the clarity. And Janet had some beautiful publications of her 340 in development, and just did really seminal mouse developmental stuff, and then moved on. The disruptive technology of mirroring genome editing, its earlier beta was becoming launching internationally, and Janet more or less steered the international group, which got the actual various knockouts together. So she moved beyond her organization to the globe and did a really seminal job in moving that forward. Crossed University Avenue to do more or less the same with Sick Kids Research Institute, really continuing its premier place. And Martin Osborne, I don't mean to, but just making it just go that much further up, a role model for us institutionally. And now has gone on to accept the Gardner, which is just great news, which means, but you won't be able to head the CIHR. So that's a bit of, we're a bit sort of ambivalent about you doing that. But just a remarkable force within the Canadian landscape, scientifically and leadership. So we're delighted to invite you to speak, Janet. Brief enough?
Thank you. Thank you, Alec. Thank you, Orby, for the honor of being here to give this talk. I am back at the University of Ottawa, having stood here introducing Jennifer Doudna as one of this year's Gardner International awardees. So now I'm on the other side of the fence, as it were. So what I'm going to do today is tell you a very little bit about my research, but I want to spend more time on why the kind of research that I've done over the years on a tiny little embryo has had huge ramifications that have led us into a whole load of ethical challenges in human health. So here we are. This is the tiny little embryo that I have spent a great deal of my research career working on. It's the blastocyst. This is a mouse blastocyst, and as it says on the right, this is only 4 days old. If it were human, it would be 7 days old, so not much different. It's about 100 cells, the size of a speck of dust. And for me, this is the most beautiful stage of development to study from a fundamental science point of view, because it only has 3 cell types. It makes an outer layer of cells called the trophectoderm, and then a group of cells at one end called the inner cell mass, within which there's a group of cells that are stained pink that are called epiblast, and a layer of cells on the surface called primitive endoderm. And this is a very beautiful picture because it's stained with all sorts of antibodies to genes that specify the different cell fates. When I started with it, we didn't have any of that. So it was just an embryo we looked at in the Petri dish. And I've spent on and off, and certainly doing other things along the way, most of my career coming back to the fundamental questions shown here. What are these 3 cell types? What do they give rise to? When are they restricted? Are they really restricted lineages that have only a specific fate later in development? How do they get established? So it's 4 days old. How do you go from an egg, a single cell, to making 3 cell types? And then, importantly, and that's the stem cell piece, can we derive lineage-specific stem cell lines? This is a tiny little embryo. We can't get a lot of them. If we could get permanent cell lines that we could maintain in culture that remembered where they came from, remembered their fate, then we'd have great tools to study early development and, of course, to manipulate genes and potentially address regenerative medicine. But now we are definitely running ahead of ourselves because that wasn't even on the cards when I started. What I spent my first little while, whoops, we skipped one there. What I spent my first little while doing as a graduate student was working with Richard Gardner, who was one of the founders of mouse chimera research. And it was he who first showed that you could take cells from one embryo and inject them. Here's a blastocyst shown attached to a suction pipette here. And what we're doing is injecting cells from one embryo into a blastocyst, having them incorporate back in the inner cell mass. And then if we had the right markers, in the first place, just coat color markers, and increasingly over time, more and more detailed in situ markers, we could then follow the fate of those injected cells in this one integrated host chimera. A chimera then, because it's an amalgam of more than one organism, like the ancient chimera of myth. So here we are, putting cells into the blastocyst, making chimeras. And variations on that theme has allowed us to indeed address very clearly the first question, which is what do all those cell types do in the embryo? Because we can reconstruct the embryo and change cells around and study their fate and find out when they're committed. Without going through any of that research, which is still ongoing, I can tell you that those three cell types are restricted lineages by the time you see them there. They can't change their fate. You can take them out. They won't. They just stay the same thing. And the outer cells, the trophectodome, give rise to the placenta, which was pointed out by Henry Friesen at breakfast to probably, I would say, be the most important organ in your life, because it was the placenta that kept you going in your mother's womb. Without the placenta, all the rest of this can't happen. Anyway, trophectodome, first cell type, makes the placenta. The green cells make yolk sac, another sort of extraembryonic cell type, both of which the mouse embryo and the human embryo needs to survive in the uterus. And it's only these little pink cells here, the epiblast, that give rise to the entire fetus. So those early chimera experiments told us that epiblast cells are the pluripotent cells of the embryo, and they can make the entire mouse. So they are pluripotent. They are not totipotent. Totipotent would mean they could also make the other cell types, and they don't. So when we talk about pluripotent stem cells, it's because they really resemble the epiblast and have a somewhat restricted cell lineage. They don't make placenta, they don't make yolk sac. And over the years, since those initial developmental studies, of course, people have gone on to derive stem cells from the blastocyst. And in the mouse, the most famous stem cells are, of course, embryonic stem cells, which come from the epiblast, and they grow in culture in undifferentiated endlessly, but they remember where they came from. They express transcription factors typical of the epiblast, and if you put them back into the blastocyst, make a chimera, same way I showed you right back at the beginning, put them back in the blastocyst, now we've got better markers, this is beta-galactosidase, all the tissues of the fetus, including the germ cells, can be mixed, host and donor, but they don't make placenta, and they don't make yolk sac. So they are pluripotent, not totipotent. And then my lab over the years has shown that you can make stem cells from the other two cell types. We have trophoblast stem cells, which grow in culture, different culture conditions, different transcription factors. They remember where they came from, put them back in a blastocyst, they contribute to the placenta and not to the fetus. And then finally, of course, Zen cells come from the primitive endodome, and they contribute to the yolk sac, but not the fetus or the placenta. So it's a very nice system that we have to explore the molecular underpinnings of early development. But really, of course, I may love the placenta, and I even love the yolk sac. Most people and most attention that's happened since the ensuing years has, of course, been around embryonic stem cells. Because these cells with pluripotency in the mouse, what suggests then that they could have enormous powers, because we can grow them indefinitely in the mouse, and yet they can, in this chimera, apparently make all cell types. So we wanted to ask the question, are these ES cells truly pluripotent? If they are truly pluripotent, they should be able to make a whole mouse, because the epiblast gives rise to the entire fetus. So this was a wonderful collaboration with my long-term collaborator and friend, Andras Naj, at the Lunenfeld, when we were at the Lunenfeld in Toronto. And we are here shown in 2013. This is, if anybody's been to Hong Kong, Ozone is a very, very high skyscraper with a bar at the top. So I think Andras is suffering from the height. I am obviously enjoying myself. So what we did a genetic trick. We tricked the embryonic stem cells to take over the fetal compartment by combining them with tetraploid embryos, which are made You don't need to know how, but it's fairly simple to make embryos tetraploid. And we knew from the literature that tetraploid embryos can make placenta and yolk sac, but they're not very good at making the fetus. I already told you that ES cells can make the fetus because they like epiblast, but they don't make the placenta and the yolk sac. So if you put them together, it's a complementation system. If this is going to survive at all, then what will happen is the ES cells will take over the entire fetal compartment. And that's what we showed initially. We can get fetuses like this, derived from these blue ES cells, and again, the placenta is green. We went on with better ES cells, and over time, this has been done by many different groups in many places around the world, and you're able to show that you can actually generate live mice that are entirely ES derived. And here they are now from a GFP line. These little litter of pups are all entirely ES derived. They will grow up, they'll be perfectly happy, they can breed. And I still, myself, find this the most amazing experiment we ever did. The thought that you can grow cells in culture for many generations and still make a whole mouse is pretty amazing. But this then was this is a highly cited paper, and I think it's cited because it really did demonstrate that ES cells are truly pluripotent, leading to the question, well, if mouse ES cells can make a whole mouse, what if we can make human ES cells? They could really be an amazing source of cells that could give rise to every cell type in the body, not necessarily in a chimera or in an embryo, but in a controlled manner to treat degenerative diseases. And of course, that came about when Jamie Thomson in 1998 was the first person to isolate human embryonic stem cells from the blastocyst, just the same way that we would have done in the mouse. And human embryonic stem cells have this amazing therapeutic potential because they really are the only stem cell that you could grow endlessly, endless numbers of in the petri dish, and be able to make potentially every cell type you might want to think about to study disease or treat disease. So the therapeutic potential is huge. So human ES cells opened up new vistas in regenerative medicine, built on this fundamental embryology that we developed in the mouse. But here's where I'm going to take a sidestep. You've heard, I've given you sort of a little run through of the background of the science. I'm now going to take us down the ethical wormholes that have opened up as we've gone into human ES cell work and human embryo work in general. So human ES cells really opened up, obviously, new vistas in regenerative medicine and new ethical concerns. So when human ES cells were first derived, it was extremely controversial. Newspapers were all over this. The parliament here, the government, certainly the government in the States and around the world were really concerned about whether we were crossing an ethical divide, because to make embryonic stem cells, you had to use human embryos. And for those who believe that all human embryos should be protected and allowed to develop into babies, then obviously this is a concern. So where did these human embryos come from that were used to derive the original human ES cells around the world, including in Canada? They were derived from in vitro fertilized embryos that came from IVF clinics. And essentially, when IVF takes place, usually more embryos are made than are needed for reproductive purposes. So with informed consent of the gamete donors, it's possible to take those embryos and use them for research, including stem cell research. But in fact, around the world still, the legislative restrictions around the use of embryos in this regard still varies considerably from place to place. And in the US, it is still, you are unable to use federal funds to derive new embryonic stem cells. You can use federal funds to work with approved existing cell lines, and you can't do any human embryo research at all. However, different states in the US, for example, have different rules. Some of them allow generation and use of embryos for research and stem cells. Some of them ban it completely. It's a complete mosaic. And you can have a map that shows variation from country to country. So the point is, what do we do in Canada? So where did Canada come into this? Well, again, cutting a very long story short, for those of you who were around at the time, there was a very extensive debate at all levels, particularly here in Ottawa on the Hill, around the use of embryos in research. And it got tied up and combined with the development of what's called the Assisted Human Reproductive Act in 2004. And this is a federal law, criminal law. There are criminal sanctions under this act. It was designed largely to deal with issues around assisted human reproductive technologies, surrogacy, egg donation, et cetera, but because it was going through at the time when embryo research and stem cell research was in the news, and as we'll see in a minute, cloning was in the news, it really became a focus of attention around the clauses in the Act that would really address the use of human embryos in research and stem cell research. And there were many, many discussions and many compromises that took place in order to bring this Act through to fruition. It does restrict human embryo research in Canada with criminal sanctions. It does, however, allow research on existing ES cell lines, and under oversight and regulation, it does allow derivation of new ES cells from IVF embryos. So the starting position is not too bad if we're talking about generating human embryonic stem cells. And while the Act was going through and the regulations that were supposed to come out of the Act were being considered, the Canadian Institutes of Health Research, I'm missing a research in there, CIHR, really ahead of the game, brought together a task force to look at the guidelines for human embryo stem cell research in Canada. And that's the task force that I chaired under Alan Bernstein when he was the head of CIHR. And that task force came up with a set of guidelines that sort of extended upon what was banned in the Act and really provided an oversight for the allowed research around embryonic stem cells that was allowed under the Act. So there is now an oversight committee called SCOC at CIHR that has to approve and does look at and approve studies using human embryonic stem cells. And I think that stem cell oversight committee has functioned quite well. It's looked at new cell lines, it's decided which ones are appropriate for use, and it's looked at the kind of studies that people want to undertake and approve them where appropriate. However, I had a little bit of fun in preparing this talk because I went back and read the Act. You should always go back and read things. And asked, well, actually, what is banned under the Act? And if we look at where science in this whole area of embryo research, gene editing, chimera research is moving today, what are the implications for those bans? And what are the implications for research in Canada? So remember, this is a criminal act. Anything I'm going to show you next, you could be thrown in jail for. So please, please don't do this in your basement. Number one thing you should not do is clone. So you should not, no person shall knowingly create a human clone by using any technique or transplant a human clone into a human being or into any non-human life form or artificial device. Using any technique, that means you cannot develop the technology for human cloning. It's intended, I believe, to deal with making human clones. Remember the Raelians? This was also part of the discussion at the time. But it also essentially prevents any kind of therapeutic use of cloning technology. So how did this come about? Well, look at the timing here. Dolly the sheep, 1997. Human embryonic stem cells, 1998. There was a conjunction of these things going on at the same time, and the discussion got kind of muddled up. But essentially, cloning was designed in the sheep to be a methodology to allow genetic manipulation of farm animals. But it also demonstrated very nicely in a mammal that the DNA, the genetic information in an adult cell, was still capable of reprogramming the whole of development. So what was shown with Dolly was that a somatic cell nucleus, in the case of Dolly, from a mammary gland of a sheep, was put into an egg in which has had its own DNA removed, and then that oocyte was able to grow up into a blastocyst, first of all, and then the blastocyst, if put back in the uterus, was able to develop into Dolly the sheep. But that was one approach, and that's what people really didn't want to see happen with humans. And I don't think anybody really is seriously looking at cloning humans in this reproductive sense. But it got caught up with what was at times called therapeutic cloning or somatic cell nuclear transfer. And therapeutic cloning would say, why don't we do this from adults, from patients perhaps, who we want to study a disease, or patients that might require a stem cell transplant. We could take one of their nuclei, cell nucleus, put it in a donor oocyte, make a blastocyst, and then make stem cells from it. So now we've got cloned stem cells that would be genetically identical to the person who gave the cell in the first place. And so that would mean that you would have personalized stem cells. And at the time, there was quite a lot of interest in doing this because it seemed to be the best way to develop new ways of studying disease and also potentially for personalized treatment. So it turns out to be quite difficult, but in the succeeding years, elsewhere in the world, because it's banned here, it has proven possible to do this and derive human embryonic stem cells from these cloned embryos. And of course, development of the technology of cloning has, as you'll see in a minute, been used to treat mitochondrial disease. So that's one of the things that is banned and still banned today, and certainly has some uses, as we'll see in a minute. However, although you might say that's a constriction or a restriction on what we might do, the game changed, and the game changed around this sort of therapeutic cloning aspect with the discovery of induced pluripotent stem cells. So this is the technology that was developed by Shinya Yamanaka in Japan, 2006, so we're jumping ahead now. No embryos are used in this approach. First in mouse and then in human, Yamanaka was able to show that he could take an adult skin cell and not take its nucleus and put it back in an egg, but actually just reprogram it by adding a set of transcription factors that are required for epiblast pluripotency. Put those into those cells, grow them in the right conditions, and you could essentially turn back the clock and turn a skin cell into an embryonic stem cell. This has been revolutionary because this is now applicable to anybody. I could go around the room and we can make iPS cells from everybody in the room, and I don't need your skin cells. You can just give me a little bit of blood, and in fact, people in China have been using urine. So if you'd like to leave a sample on the way out, we can make your iPS cells. So, but what does this do for us? It really does mean that, of course, we can model human disease because we can take patients who have diseases, progressive diseases, that we can really model their development in the petri dish. That allows us to do drug screening. It allows us toxicity tests for known drugs. And it does potentially, again, allow this rejection-free transplantation, personalized treatments. So you have to be able to take these cells and differentiate them into the right cell types. And that's the big challenge that everybody is undertaking today. So that was a rather important discovery. And importantly, the Nobel Prize for Medicine in 2012 went to this great pair here, so 40 years apart, John Gurdon, who is the person who first did cloning in frogs, and then Shinya Yamanaka, who really reprogrammed adult cells. And I like to put in the Gairdner plugs. Here's Yamanaka receiving the Gairdner Award in Toronto, me. And this is 2009, and it is one of the goals of the Gairdner Foundation to be ahead of the curve. And we were ahead of the curve on this one. So pluripotent stem cell therapies, both embryonic stem cell and iPS-derived, are actually moving to the clinic. It's moving fast. It's not easy. There are safety issues. There are differentiation issues. But we are seeing some of these innovative clinical trials coming forward now with human embryonic stem cell and iPS cell-derived cells. So spinal cord injury was one of the first ones to the clinic, funded by the company Geron, which then dropped it, and it's now being picked up by another company. This is to use ES cell-derived cells to remyelinate and help to potentially alleviate damage in spinal cord repair. Perhaps the most promising one is a kind of odd one in a sense. It's making retinal pigment epithelium from embryonic stem cells, which turns out to be quite easy for reasons that escape me. But it does mean you can get a very pure population. and it's being tested for macular degeneration with both ES cells and iPS cell-derived cells underway, actually in several places around the world. Parkinson's, another good target. You can produce dopamine-producing neurons, and there are already fetal transplant studies and so on that show that it's possible to have some alleviative effect on Parkinson's by replacing these cells. And there's a very large international consortium really moving very fast towards phase 1 studies with this. Diabetes, can you produce insulin-producing cells? We know here in Canada, we have the Edmonton protocol for islet transplantation. We know how to treat it if you can get the right cells. And again, there's a big push worldwide in doing this, and there are the first trials underway, including some in Edmonton. Heart disease, this is one where there are many, many stem cell trials, but there are none yet using embryonic stem cells. But around the world, again, 2 or 3 groups, including a very active group in Toronto, are really have generated very good heart muscle cells from ES cells and have very exciting preclinical data. So we'll see this one moving forward in the future as well. So it's early days, but it's moving along. So having embryonic stem cell research, having iPS cells gets us forward, and there's certainly lots to be done in this area. But what else do we get stuck on when we go into the AHRA? Here's the next one. They're not in order. I've sort of shuffled them around a bit to make my story, but this is F. Alter the genome of a cell of a human being or in vitro embryo such that the alteration is capable of being transmitted to descendants. That is germline manipulation. If you change the genome of the human embryo in such a way that the embryo, when it's born into a person, carries the alteration in their germ cells, in their sperm or their egg, and can carry it into the next generation. So this is germline manipulation. And a lot of nervousness over many years around this concept that if you manipulated the embryo, would we be enhancing human beings? Would we be really manipulating and changing what it means to be human? So there's a lot of ethical and as well as practical considerations around this one. And it's right back in the news again today. So first of all, this ban in the AHRA actually bans mitochondrial gene replacement therapy. Mitochondria have genomes. There is now a technology for people who inherit mitochondrial diseases that are encoded in the genome of the mitochondria, that you can have this situation where the mother's egg has these unhealthy mitochondria, and if you were to just fertilize this in the normal way, all of the mitochondria of the offspring essentially are inherited from the mother. So because the sperm is a tiny piece of this, it contributes its nucleus, but it does not contribute the mitochondria. Mitochondrially encoded diseases are maternally inherited. So the concept here is to treat mitochondrial disease by essentially taking out the nucleus of the mother's egg, taking it and putting in it a donor egg that has healthy mitochondria, take away its own nucleus, put in the mother's nucleus, and then fertilize with the father's sperm. So you've got this is now genetically related to the parents in terms of the nuclear genome, but the mitochondrial genome is inherited from the donor egg. Hence, you'll see it referred to, again in the popular press, as 3-parent babies. Now, if this baby is female, then the mitochondrial genome will be inherited into the next generation. So you have changed the mitochondrial genome that would have been normally the case when you did normal fertilization. So this is considered as germline editing. If this baby is male, it doesn't go in into the next generation. So where are we at on this? The first 3-parent baby was just reported in the press in Mexico, which always makes you a little nervous, but there it is. We await the proper scientific publication on this. But the procedure exists. It's doable. It's been done in preclinical models, and there's no reason to think it can't be done in the humans. And it has been approved in the UK as potentially a treatment for mitochondrial disease. It's not yet been performed. In the US, the FDA, which has to approve these kind of genetic manipulations, has been banned by Congress from considering any protocols. So it's not going to happen in the States. At the same time, the FDA asked the National Academy of Science to set up a task force which spent a long time looking at this and came down with, I thought, a rather cop-out recommendation, which was that maybe it's okay to do, but you could, if you did it, you'd have to only transfer back male embryos, which seems like a gender discrimination, but it, again, deals with this fact they didn't want to go down the path of essentially approving germline genetic manipulation. I think, and again, we would need a clearer guideline from Health Canada, but it seems to me that this is probably totally banned in Canada, although perhaps, again, restricting to the male embryos might be considered okay. I just don't consider that's ethically acceptable. So there's one that's potentially on the list of difficulties, given what we can do today. But really, this clause was not really designed to hit about the mitochondrial genome, although that one's right there, sort of ready to go as we stand. But it was primarily directed towards altering the nuclear genome, and really was dealing with both correcting genetic disease, which most people might consider is okay, but more concerned with this idea of genetic enhancement, making a super race somehow or other. So what can we do in terms of rewriting the DNA of the early embryo? And actually, Alec mentioned that part of what I did with the mouse stem cells and with early embryos is develop a lot of the technologies for genetically altering mouse embryos. So techniques like transgenesis, where we can stick DNA into the egg, or targeted mutagenesis in embryonic stem cells, where we can make a mutation in stem cells and then put it into a chimera and have it transmit to the germline. They've been around in the mouse really since the 1980s. And they all, of course, allow gene alterations to be inherited in the next generation. That's the point. We want to make genetically altered mice. We want to use them for studying gene function. And we want to use them to generate mouse models of human disease. And it's been a big push and continues to be a big push in mouse genetics. And so the technology has been there, and people have thought about this, what happens if you do it in humans, for a while. But none of the technologies that we use in the mouse were really efficient enough that you could even think about applying it to a human embryo. So it was always easy to say, well, yes, no, yes, I know there are ethical concerns, but it's just not feasible, it's not safe, we wouldn't even go down that path. Then things changed, another game changer. So this game changer is one that if you were here a week and a half ago when I was here with Jennifer, you would have heard all about. This is CRISPR-Cas9 gene editing. And this is not fundamentally different from anything we've been doing over the years in the mouse, which is to genetically rewrite the DNA within an egg or a cell, but it does it using a gene editing system that is much, much more efficient and highly specific. It's a gene editing system derived from bacteria, and it can target a cut in the DNA at almost any site across the genome in a very specific manner, and also help promote replacement at those sites. So it's very specific, very efficient, and again, it can now be used and is being used to genetically modify plants, animals, a big agricultural impact, bacteria themselves, fungi, and of course, we use it in the mouse all the time to make new models of human disease. And it's opening up a whole new era, bringing back gene therapy back really to a new level in humans, where it's possible, again, because of the efficiency, to be able to genetically correct disease like sickle cell anemia, beta thalassemia, in cells, in hematopoietic stem cells, and think about putting them back in patients, treating cancer, the development of CAR T cells, chimeric antibody modified T cells, also uses gene editing technologies, preventing HIV, and work in the mouse would suggest that you might even be able to do this to treat something like Duchenne muscular dystrophy, which is a very widespread disease. But again, the efficiency, better ways of getting the vectors into the cells and the organisms is coming, and it's coming very fast, and it's basically impossible to keep up. So here we go, another little plug. Here we are at the Gairdner Awards. So again, ahead of the game, the Gairdner Awards recognize this whole CRISPR-Cas9 gene editing revolution a couple of weeks ago now. And this is what I call the CRISPR-Cas quintet, these 5 people in the middle here, Feng Zhang, Emmanuel Charpentier, Rodolphe Barangu, Jennifer Doudna, and Philippe Horvath, and more people beside, have really been the people who've driven this revolution forward. So, fine, it's got lots of good things you can do with it, but it does now raise the issue that it is efficient enough that you could think about doing this in a human embryo or in gametes. So what happens? What would you do if you were to genetically edit the human germline? What's acceptable and what's not? So this is just briefly, this is what happens when CRISPR-Cas9 comes in. Here's the host DNA. There's a guide RNA that is complementary to a particular sequence in the genome, causes the DNA to separate. This nuclease comes in and cuts, essentially, at this very specific site. You can introduce new genes, you can cut out genes, you can make point mutations, you can do a lot of things with this approach. So you could think about making this little baby cured of cystic fibrosis or Huntington's disease by genetically manipulating the embryo or the gametes. You could perhaps take a little bit, and most people would not think that was a bad thing to do. There are practical issues, which we could discuss later, but on the face of it, curing devastating single gene diseases, which could be done, seems like a reasonable thing to do. You might think about perhaps something that's moving towards the enhancement side. Should we address some of the genes that predispose to Alzheimer's, breast cancer, or strokes? Could we make humans less likely to have Alzheimer's? We know some of the predisposition genes there. We could alter them. Is that acceptable? And then, of course, you move on to these other things that make people more nervous. That is to say, could we make better muscles? That's a good one for around here, I think. High IQ, 20/20 vision, whatever you fancy. Now, of course, the bottom line is we don't know a lot about the genetics of many of these things, so this is a long way off. But there is an issue around where you draw the line. It's not an easy thing to decide. The simplest way is to do what Canada has done and said, nothing. You can't do anything. Then you don't have to consider the ethical boundaries. But this has been and is an ongoing discussion around the world. There have been many organizations and agencies that are considering the ethical boundaries. One of the areas that's most close to my heart is that with gene editing, we can potentially study human embryonic development and begin to understand a bit more about the fundamentals of how a human embryo develops in the same way that we've been able to understand it in the mouse. And most of the discussions that have taken place around the world have come to the general consensus that the use of gene editing to study the biology of the human embryo in a petri dish is probably okay. It really is. You're not going to transfer them back. It's not going into the germline in that sense. And it would allow you to really study and understand the differences and similarities between mouse and human, and understand better how to make IVF babies, understand infertility, and also understand better how to derive stem cells. It's not clear to me in the Act as it stands whether that would be allowable or not. The wording is a little unclear, whether if you don't put it back into a mother, then whether you'd be allowed to do it. It's not clear, and it's not clear that we have a clear regulatory environment in which to do this. It seems to me this is something potentially for CIHR to take up. This approach has been approved in the UK, Kathy Nyakin at the Crick Institute, and my ex-postdoc, Frederick Lanner at the Karolinska Institute, has been approved to do gene editing for fundamental development in the Petri dish in Sweden. Lots of attention on this, and like everything else, I always get sucked into these debates. And here, there was a very large international summit on human gene editing at the National Academies at the end of last year. And that whole group looked at all sides. It was a very good meeting. We heard from patients, we heard from clinicians, we heard from scientists, we heard from the public, we heard from policymakers. And at the end of that, this is the sort of recommendation that the organizing committee came up with, which basically was interpreted as a moratorium, saying, let's just hold off. We're not sure yet whether it's safe and efficient, so you really don't want to do this and do it wrong. And that's true. And there is not, and I would agree, there is no broad societal consensus about the appropriateness of the proposed application. And of course, clinical use has to have a regulatory oversight. And at present, none of these criteria have been met. So that was put forward in the American and international, because this was a consensus, international global discussion. Everybody felt that essentially around the world, we did not have a clear environment in which to move forward. So this is still up for discussion, and there have been some beginnings of debate here in Canada. The Stem Cell Network had a workshop a little while ago. CIHR has a discussion document coming forward on this. So we're moving forward, but I think this is an area where, again, we probably need to have a much more broad societal interaction and discussion on where or when and how we should draw the limits. Okay, gene editing. You might think we've finished. No, we haven't finished. What else does the AHRA say? It says you can't create an embryo for any purpose other than creating a human being. Okay, so we can create a human being even if we created it in the petri dish. And we need to create some embryos, and it's true, IVF programs obviously need to do some experimental work to improve IVF and to provide instruction. So you're allowed to create an embryo and not put it back and just for instructional purposes. But you're not allowed to create an embryo essentially for research. That was designed around exactly what I just said, not creating embryos specifically to use them for, say, stem cell research. But it opens up an interesting question about what would happen if we could create an embryo in vitro artificially. We would actually probably be obliged to create a human being from it. So have a think about this one. There is rapid scientific advances going on in the world in generating eggs and sperm directly from pluripotent stem cells. And I'm not going to go through all the science of this. It's particularly being led by outstanding research groups in China and Japan. And they really are interested in understanding how germ cells, how the cells that give rise to the eggs and sperm are set aside in development, and what are the processes that lead to their formation. And in so doing, they've been able to, in this case, again, only in the mouse at this time, they have been able to take mouse embryonic stem cells and take again, starting from the blastocyst, this would be the normal way that germ cells develop. You make an early embryo, you make a fetus. The primordial germ cells arise in the early testis. They make spermatogonia, spermatocytes, and spermatids. And you can fertilize an egg and make a mouse. What they did was to take embryonic stem cells, make them into sort of cells like the early embryo, turn them into precursors of germ cells, and then by providing some support cells that resemble the cells of the testis itself, in culture, these cells were able to mature, go through meiosis, because sperm only have half the DNA, and they could get cells. They weren't swimming sperm, but they don't need to be, because there's a process called ICSI, intracytoplasmic sperm injection, where you can take a developing sperm cell and inject it in an egg, and they were able to make viable mice. Just very, very recently, really, really recently, just a month ago, less than a month ago, Hayashi's group in Japan have been able to do the same thing for the female germline. They were able to take embryonic stem cells, actually iPS cells, so they took skin cells, turned them into iPS cells, and were able to mature those cells into oocytes in culture. So these are oocytes derived from embryonic stem cells. So the whole cycle of the female germline. And again, by fertilizing these eggs, not all of them, it's not 100% efficient, but you were able to get mice. So again, quite remarkable. Nobody has yet put the two together, but it's a kind of obvious next step, in which case you would be creating an embryo. You would definitely be creating an embryo. And it's not yet been done with human stem cells. And that's partly because the process for forming germ cells in the human is different. So it's one of these areas we need to understand more about. But presumably, science being what it is just a matter of time. And there's a lot of interesting things that come out of making gametes in the petri dish. A lot of good things, but again, if you think about it, a fair number of ethical concerns. Okay, haven't finished. What else does AHRA say? No person shall knowingly maintain an embryo outside the body of a female person after the 14th day of its development following fertilization or creation, excluding any time during which its development has been suspended. And this has been around for a very long time, from the very early days of being able to make IVF babies. The biggest sort of newspaper story was, we're making test tube babies. My goodness, we can fertilize an egg, we can make a blastocyst. It's only a matter of time before we have little babies in a test tube. So to sort of deal with that, and again, dealing with the fact that nobody was anywhere near developing any culture conditions that would allow such development to occur, the world essentially accepted this 14-day rule as it's become known, which really marks in normal human development the time when the body axis and the nervous system just starts to be formed. And again, it's easy to agree to it and agree to it being in the act because there were no culture systems available to think about growing embryos over that time period until this year. And just this year, two groups published not perfect culture conditions for sure, but culture conditions where they were able to take human embryos and grow them from, this is a blastocyst here with the inner cell mass, attached in culture, trophoblast spreads out, and we start to see forming in the middle here a little embryo with a cavity that's going to go on to form this structure here, which with a little imagination, as shown in this diagram, you can call a developing embryo with an amnion, the epiblast, trophoblast, and the yolk sac. So it's on its way to undergo what we call gastrulation. Gastrulation takes place whereby these epiblast cells make all the 3-cell germ layers of the embryo and start to make the body axis. So this has also then opened up a lot of discussion. Are we approaching the ability to undergo gastrulation in the dish, make the primitive streak, begin to form the body axis? And is this a line that we should not cross? Is the 14-day rule inviolate? And again, here's a normal embryo. This is how it would look. The primitive streak is going to form here in this epiblast, and you're going to start seeing that this will be the back end of the embryo, this will be the head, and things begin to move forward. These cultures that they have are still not perfect, they're kind of flat, but you're getting close to the time when you should see that occurring, and also to the time actually where you make germ cells. So that's where the cultures are moving. Why would you want to go forward and actually study human gastrulation in vitro? Is it okay to say we don't want to cross that line? And again, I'm not sure I know the answer to that. But essentially, sorry, I popped out of it. Sorry, that's my fault. Sorry, just a second. We'll be back in a minute. We'll be back after these messages. So there are some reasons why you might think about this, and certainly fundamental understanding of a key stage of human development, inaccessible in any other way. Right? You cannot get at human embryos at this stage in the intact environment, obviously. And for those of you who are just interested in sort of geekdom, the Radio Lab has a really nice podcast on the primitive streak. I think you should listen to it, because what it is, it just says, God, it's so exciting. These people are actually looking at the primitive streak in a dish, understanding where we come from. That is a fundamental joy of doing research. So there is some fundamental interest in understanding where we come from. But it's also, how could we better understand stem cells? How could we understand germ cells and development of infertility? Modeling the implantation process, understanding how the placenta forms, and the reasons for what is a very high early embryo loss in humans. And again, if you combine this with CRISPR gene editing, you could study the genetics of early developmental defects. So there are some interesting approaches if you could do this. Perhaps we didn't use human embryos, so can you actually use if we're worried about using human embryos and crossing this line, maybe we don't need human embryos. I've just told you can make oocytes and sperm from stem cells. Can we make an organized embryo from stem cells? So there's been a couple of papers showing that if you take human ES cells and grow them in what's called a sort of micro-patterned culture on a right substrate, then these stem cells will start to organize themselves. Obviously not something that is a three-dimensional embryo, but in 2 dimensions, this embryo is organizing itself with the cell types in the same order that they are in the embryo, allowing cells to talk to each other, and again, potentially allowing primitive streak formation and body axis formation in the petri dish from the embryonic stem cells. So that says we don't use human embryos, but now raises the other question that's come up in the literature as well. If these embryos that are growing from the stem cells are starting to become organized, do they have emergent properties that themselves might challenge the 14-day rule and what it means to have human organismic potential? Last one on the AHRA. What else does it say? No person shall knowingly create a chimera, that's what I've been doing all my life, transplant a chimera, it's a human chimera they mean, transplant a chimera into a human being or a non-human life form. It's particularly because this is the Human Reproduction Act, focused on human-human chimeras. I don't think anybody has any intention of making human-human chimeras, but it's not very clear about human-animal chimeras. However, the Tri-Council policy statement, which comes out of the CIHR guidelines, is quite clear. Article, whatever it is, prohibits research in which human embryonic stem cells, embryonic germ cells, induced pluripotent stem cells, or other cells that are likely to be pluripotent are combined with a non-human embryo or non-human fetus. Full stop. So any taking of pluripotent cells and putting them into a fetus or an embryo prior to birth is not allowed. Post-birth, you can do preclinical studies with embryonic stem cells and their derivatives, but not prior to birth. This is really quite restrictive. So why would you want to do this? Why would you want to take human cells and put them in an animal? You want to make interspecies chimeras because they allow you to cross species boundaries and study cell potential. So interspecies chimeras have been around for quite a while. And I was really one of the first people to make interspecies chimeras, not between humans and animals, but between 2 species of mice. These 2 species don't breed with each other, but if you take inner cell mast cells from muscaroli and put them in a mouse musculus blastocyst, you can make perfectly normal mice. So you can extend the barrier between species by making chimeras. With that in mind, then, could you go further and could you actually extend the species boundaries enough to be able to get human cells to incorporate in a meaningful manner in any animal? So this has got a new lease on life, largely through the work of Hiro Nakauchi, first in Japan and now at Stanford, and who has done a sort of proof of principle experiment, moving forward to think about generating organs of one species within the animal of another. So in this situation, what he did here was to take I'm so close to it, I'll have to read it off here, you'll have to follow it yourself. He took a mouse that is genetically unable to make a pancreas. First of all, he took mouse embryonic stem cells and made a chimera. This is now going to have mixture in all its tissues, but the pancreas is kind of empty. So rather like that tetraploid complementation where we took over the whole fetus, in this case, the embryonic stem cells take over the whole pancreas. So this generates a mouse-derived pancreas in a different mouse. So that was intra-species. He then decided to see if he could cross species. He made rat-mouse chimeras and showed that he could get mixtures of the 2 cells. So then he took a PDX mouse and took rat embryonic stem cells, and now you're generating a rat pancreas in a PDX1 minus mouse. So now you've got a rat pancreas growing in a mouse. You've also got other rat cells, which is an issue. So that was all proof of principle because his vision is generating human organs in large animals, and his choice of large animal is the pig. And these experiments are being developed right now in the US. in California. And the concept here is to have a pig that also cannot make a pancreas, and then take human iPS cells and inject them into the pig blastocyst. Again, this pig will then grow up. It would have potentially a human pancreas. You could isolate the islet cells in large amounts and use them for transplantation. That's his vision. There are many blockages along the way. It's not at all clear that this is I guess it's time to stop. I know, and I'll be finished in a minute. This is like the Oscars when they turn up the music, right? Come off the stage. This pig, I can't end. I've got to go through the pig here. So it is difficult. It's not at all clear that human cells, the distance between the pig and the human is huge. So rat mouse is far enough. Mouse, we can go. Rat mouse, you can stretch it. Human to pig may be too hard to go. But there are tricks along the way that they're pursuing to try to make these cells incorporate. And I don't think we should discount it as a potential future. So in sum, the science of stem cells, of embryos, of germ cells, gene editing, chimeras, is moving apace. And clearly, these raise along the way exciting scientific discoveries, potential new therapies for humans, but the ethical, legal, public policy implications are really lagging behind, especially in Canada, because we have an act of the Parliament that is very difficult to change. So I think it is time, and I think there's an opportunity and a willingness on various parties to really start to engage a broader range of stakeholders, patients, public, policymakers, to debate how and where to draw the boundaries, and how, in Canada, we can develop a regulatory environment that, while allowing while not allowing anything goes kind of research, that allows a clear regulatory environment, particularly around these boundaries of human embryo research going in the future. So it all begins with the blastocyst. Thank you. Do I get questions or not?
No, I don't think so.
Okay. Yeah.
Thank you, Dr. Rossant. I'm always impressed at times like this that researchers and broadcasters have a fair bit in common, and I think actually it goes back to the genesis of the word broadcasting, which is not a word that has anything to do with technology or spreading things by radio waves or television signals. Broadcasting is something that involves sowing seeds, literally broadcasting. And I think that both broadcasters and researchers, scientists, have a great deal in common in that it is a responsibility that comes with the job description that when you find something that is important and interesting and potentially essential to the human race, you have a responsibility to spread that information around. And that is definitely what Dr. Rossant did this morning and does with her career. And this was, I think, a very elegant, very concise description of both the exciting possibilities of the research that she and many of her colleagues have done, but also the ethical implications that need to be considered before we go any further. At this point, I would like to invite the person for whom the Friesen Prize is named, Dr. Henry Friesen, to come forward with some comments and remarks on what Dr. Rossant has said.
Thank you very much, Paul. Thank you, Janet, for a magnificent presentation that really inspires us because of the clarity of the exposition, which really underscore that the vocabulary of science has changed dramatically. Tools of science now are ever more precise and sophisticated, and yet you have simplified them for all of us, even though few of us are necessarily in the field. Most importantly, you have confronted head-on the ethical challenges and issues that have surfaced because of the advances in technology. But you haven't simply surfaced them. You have led not only the discussion in Canada, but around the world in contributing to framing the issues and how we could move forward in a creative way in addressing them by outlining, as you have in the past, the social license, seems to be a popular term these days, that will lead to ultimate sanction and approval of the way forward. It's a very compelling research story that has emanated from your lab. The impact of these discoveries are yet to be fully realized, but I think we can, with a little imagination, glimpse the vast potential that these discoveries are likely to generate and the applications. I think, too, we should all thank you for your leadership, not only in science, but for the many institutions in Canada that you have contributed to by your leadership, as well as the public policy. You've awed us, really, by the range of your contributions, and we're so grateful, and I'm personally so grateful, and indebted to you for all of those things. Thank you very much.
Thank you, Henry. I want to add again my thanks and congratulations to Dr. Rossant for winning the Friesen Prize this year. And thank you all for coming out this morning. There is a reception that will take place immediately following upstairs and at the back of the hall. So, sorry.
Thanks abound. I won't extend the discussion further. We're all excited and delighted to have experienced your presentation. We have to vacate by 1 p.m. and if anybody's left over, you'll have to sit for an exam, because that's what's happening at 1 o'clock. But there is a final act here, and that is to present Dr. Rossant with a memento of the event, and I'll ask Alex McKenzie to come and provide this on behalf of the institution.
All right, thanks very much. I picked it out myself. Can I see what it looks like? Great talk, Janet. Brilliant. Well, it's a picture of my house in Ottawa. A memento. Okay, where we had breakfast. Okay, all right, great. Good, good.
So it's just the two of you in this, or do you want to go?
She's happily married.
You can put that down.